Ladies and gentlemen, welcome to the Shaw Prize Lecture in Life Science and Medicine 2025, hosted by Shaw College of the Chinese University of Hong Kong. May I first invite Professor Freedom Leung, head of Shaw College, to deliver his welcoming address. Professor Leung, please.
Professor Baumeister, distinguished guests, colleagues, students, ladies and gentlemen, good evening. Welcome to the Shaw Prize Lecture in Life Science and Medicine 2025. Sir Run Shaw established the Shaw Prize in 2002 to honor individuals who have made groundbreaking contributions in academic and scientific research or applications. Since the first Shaw Prize presented in 2004, Shaw College has the honor to host the Shaw Prize lectures every year on one of the three prizes, astronomy, life science and medicine, or mathematical sciences. Through presenting lecture of the Shaw Laureate to professionals, students, and the general public, we strive to uphold Sir Run Shaw's visions on the quest for knowledge, as spelled out in the founding aspiration of Shaw College, learning, innovation for benefiting humankind. I would like to congratulate Professor Wolfgang Baumeister for his revolutionary advance in cell imaging technology. We are very honored to have Professor Baumeister here today to share with us his development of cryo-electron tomography to reveal the inner working of cells. We are also grateful to Professor Byung-ho from the School of Life Sciences to introduce our Shaw Laureates in Life Science and Medicine and moderate a Q&A session later on. I shall now pass the time back to our MC. Welcome all again to Shaw College.
Thank you so much, Professor Leung. May I now invite Professor Byung-ho from School of Life Sciences of CUHK to introduce the Shaw Laureate in Life Science and Medicine 2025. Professor Kang, please.
Good evening, ladies and gentlemen. This year's Shaw Prize in Life Science and Medicine is awarded to Professor Wolfgang Baumeister for his pioneering work on cryo-electron microscopy, which is a three-dimensional electron microscopy technique visualizing cells and subcellular structures, including organelles and cytoskeleton and macromolecular complexes in their natural cellular context. So Professor Baumeister is currently a director, emeritus, and scientific member of Max Planck Institute of Biochemistry and distinguished adjunct professor at ShanghaiTech University. He studied biology, chemistry, and physics in college and got PhD in biophysics in 1973 and worked as a research fellow at Cambridge University in UK. In 1983, he joined the Max Planck Institute of Biochemistry, where he led the molecular structure biology department, served as a director till 2021. And since 2000, he also has been an honorary professor at the Technical University in Munich. With respect concerning membership and award, Professor Baumeister is a member of German National Academy of Science and a member of American Academy of Arts and Science, as well as an associate of U.S. National Academy of Science. He, including many honors, he was awarded Otto Warburg Medal and the Louis Adjunct Prize for Medicine and Rosenthal Award. So we are delighted to have Professor Baumeister with us today to give a lecture on cellular cryo-electron tomography, the power of seeing the whole picture. Let's welcome Professor Baumeister.
Yeah, good afternoon, ladies and gentlemen. Thank you very much, Professor Ho, for your kind introduction. So it's my pleasure to give the Shaw Prize lecture here at Shaw College. And for reasons which are pretty obvious, I think, I will talk about cryo-electron tomography, or cryo-ET. So it's an imaging modality which allows to do structural biology in situ, as I like to call it. Others prefer to call it in-cell structural biology. So let me first, give you a couple of reasons why we want to do structural biology in situ at all. I think normally structural biologists approach cellular complexity in a reductionist manner. Cells are opened up, the molecular contents are released, they are fractionated, and specific or subsets of the molecules inside the cell are purified, and then they are studied one by one using the established methods of structural biology, X-ray crystallography to some extent, NMR, and increasingly cryo-electron microscopy in the form of single particle analysis. Now, this divide and conquer approach has been very successful if you look at the protein data bank, but it's also clear that only rarely, let's say, cellular functions can be attributed to individual molecules. Cellular functions arise from the interactions between the many molecular species inhabiting cells. So they have to act somehow in concert. Now, I think structural biology in situ aims at studying macromolecular or supramolecular structures in their functional context inside unperturbed cellular environments. Now, I think I should begin by mentioning, a remarkable paper that was published back in 1968. So it was entitled Electron Microscopy of Unstained Material, the Polytropic Montage. Now, it was published by a person called Roger Hart. He was at Lawrence Berkeley National Laboratory. And if you read the abstract, saying with the use of an electronic picture scanning device and a digital computer, micrographs taken of a specimen along several different directions can be superimposed to form a montage that is more informative than the component images. Preliminary results indicate that one may thus study unstained, unshadowed biological material at high resolution. Yet clearly an idea or a vision, but I should say that this paper, was very rarely cited, and it was largely ignored. Now, I think it's a typical example of somebody who had a vision, but at a time that was not yet mature. The technology needed to do that. The polytropic montage is obviously synonymous to tomography, did simply not exist at the time. There were many bits and pieces missing. So, but he was on the right track. I think he then developed the first useful CCD cameras in order to have, an electronic picture scanning device, etc. Now, but he passed away shortly after publishing that paper. So I think, but I think it's worth mentioning that I think he had a clear vision. I think his idea was certainly not to apply it to cells. I think it was at a time when people were still struggling to use electron microscopy to study or to determine the structure of isolated molecules. Now, so what has been described here in the abstract, is clearly the principle of electron tomography. So basically, one takes images under different orientations of the object by tilting the object in the beam, and these images are then merged into a 3D structure by methods like weighted back projection. I don't want to go into the details of the reconstruction mechanism. So now what is cryo-electron tomography? It is the application of tomographic principles of data acquisition and reconstruction to objects embedded in amorphous ice. So by doing that, basically all organelles, all supramolecular structures or macromolecular complex are kept at their native place as they are in the cell. I think a strict definition of in situ structural biology is really that I think everything must be frozen in as it exists in the living cell. Now, okay, that is again on the left-hand side, basically taking a tilt series, and below that you see, I mean, how things are then merged in order to get a reconstruction of a cellular volume. Now, I think there was very strong skepticism at the time we started to develop cryo-electron tomography. That was in the late 1980s. So people made actually jokes about it, saying, well, it's difficult enough to determine the structure of molecules in isolation, and now you want to do that in the crowded environment of a cell. The situation, others, I mean, had other arguments, but I think there were basically two lines of arguments against doing it. One group of people said, is it worth it? They felt, I mean, what do you expect to learn from doing cryo-electron tomography? That reminded me of the situation, let's say, in the 1930s, when Ernst Ruska was developing the first electron microscope, that was basically end of the 1920s. And I think it's worth, I mean, to read this as a part from his biography. In Berlin, at least 3 research groups were striving for technical progress in the construction of electron microscopes. In the mid-1930s, the situation entered a critical phase, since many influential biologists and physicists doubted whether electrons could be used in biomedicine for high-resolution imaging. The ability to visualize sublight microscopic structures, if indeed they existed in the living nature, was seriously questioned because a state of continuous change was envisaged, which would in principle prevent these minute structures from being visualized. Even assuming the existence of well-defined supramolecular structures, it was thought that the electron microscope and the necessary preparation techniques would be incapable of furnishing a correct structural analysis of biological objects. The fine structure would be completely destroyed by dehydration of the biological sample in the vacuum of the instrument, and would in any case be burned to ashes in the microscope's intense electron beam. And I think similar arguments were put forward when we started to do cryo-ET. I think this is a very nice anecdote, also illustrating the point made in the previous statement. It's from a conversation between 2 eminent scientists, I mean, Leo Szilard and Dennis Gabor, who won a Nobel Prize for inventing holography. You know, Szilard taught Gabor in a cafe table in 1927, now that it is possible to make electron lenses, why do you not make a microscope with electrons? At smaller and smaller wavelengths, you would achieve much more detailed resolution than is possible with microscopes using light. Gabor and Szilard pondered this idea for a few minutes. Then they agreed it would serve no useful purpose. After all, you could not put living matter into the kind of vacuum tube needed to control electron beams. Besides, they concluded so much power would be focused in the electron beam that it would incinerate any sample. So, I mean, basically 2 arguments. First of all, I think in the vacuum it will become dehydrated, and I think biological structures need to be kept hydrated, and that problem was only overcome with going from to frozen hydrated samples. What are the problems? I think they had a point in what they said. I think the problem in electron tomography, and that was, I mean, an argument that it might not be possible to do it at all, was to obtain a detailed and undistorted reconstruction. A tilt series must be recorded that covers as wide an angular range as possible in as many increments as possible. So you see here, if you have, let's say, only 18 projections and 10-degree increment, I think the reconstruction in this simulation, I think, does not look very good. If you go to, let's say, 72 projections with increments of something like 2.5 degrees, you get a fairly well-defined reconstruction. So I think you would like to maximize the number of projections, but at the same time, in order to keep the electron dose acceptable, you need to minimize, the exposure to the electron beam. And that is obviously difficult to reconcile. The only solution comes from a physical principle called dose fractionation. Basically, it means taking the maximum allowable electron dose and distributing it over as many projection images as possible. The consequence is, of course, then the images become increasingly noisy. Now, you can do that only to the extent that you can still align the images for the reconstruction by cross-correlation. You see in the middle panel, there is still a very well-defined correlation peak allowing the alignment. Yeah, I think the second element that was needed, and that was also, kind of mentioned in Roger Hart's little paper, you need automation because, there are many steps you have to correct for. Like goniometers don't work with the precision of, let's say, a few nanometers. So, one had to really, automate the data acquisition. And that was even in the late 1980s when we started a major challenge. I think CCD cameras became available, and we were lucky in a sense that one of our graduate students had a startup company developing and improving CCD cameras. But we needed also software and hardware, for many auto-tuning functions, autofocus, etc., in order to keep the dose low. Now, this was historically our first automatically recorded and reconstructed tomogram. It was something as just a piece of dirt sitting on a carbon film, so probably a little bit of carbon, but it convinced us, yes, we can do all the auto-tuning things. And I think a little bit later, that is the reconstruction, but nobody cared for the reconstruction of a piece of dirt. Now, one should say that with many auto-tuning functions like low-dose procedures and autofocus, etc., we could show then, that was 92, that the total dose or the cumulative electron dose in collecting tomographic 3D datasets can be kept small enough for the investigation of ice-embedded specimens. So I think the basic problems of tomography at that point in time had been overcome. Then I think that is the first cryo-electron tomogram we ever published. That was back in 1995. Again, something fairly trivial, lipid vesicles embedded in ice. Then, in order to develop image processing techniques further, we began to use these vesicles as kind of phantom cells, filling them with, in this case, actin. Let the actin polymerize inside the vesicles, stretching out the vesicles, and then I think we can do a reconstruction. You see on the left-hand side, because of the dose limitations, the reconstruction, even though, when we combine all these noisy projection images, signal-to-noise ratio improves and builds up again, is still fairly noisy. And we began with the development of what is referred to and widely practiced today, denoising. Denoising is basically a sophisticated way of low-pass filtering, eliminating the high-frequency noise but maintaining the signal. It improves things, but it is still not a perfect solution. Now, going from vesicles to small objects at the time, that was relatively easy. So I think the next step was, to apply it to virus particles. That was the work, done by Kai Grünewald in the lab. That was herpes simplex virus here. And what you see on the left-hand side is projection image. On the right-hand side, you see slices to the reconstructed 3D volume. And you see, the improvement in seeing fine detail. Now, again, to deal with the noise and to suppress the noise, more powerful than denoising is averaging. So if we have repetitive features in our structures, we can extract them computationally, align them, and we can average them. And then I think we have a much improved structure, as you see in light blue, nucleocapsid here. That is a symmetrical structure, and so we can easily do averaging here. And it was the first example of subtomogram averaging at the time. Now, I think from there, we wanted to move on to cells, because that was the idea about developing all this. And so what you see here is we learned how to grow many cells directly on EM grids. This is now dictyostelium crawling on an EM grid here. And so that was our first attempt. I think there were a couple of obstacles, but like avoiding, poisoning of the cells, etc. But it worked well. On the left-hand side, you see again a projection image of the periphery of a dictyostelium cell. On the right-hand side, you see a slice through the tomogram. You see the filaments. These are the actin filaments, and nestled in between are sitting the ribosomes. Ribosomes are a favorite for people doing electron tomography because they have relatively high contrast because of the RNA content. That paper was a kind of a turning point. People began to believe suddenly that tomography could become something useful. It was also, because the journal Science, selected it as breakthrough of the year. But there was still a big problem remaining. Now, what you see here is, again, a projection image of a dictyostelium cell sitting on a grid. And you see, basically, that only the periphery of the cell is thin enough to be electron transparent. The center around the nucleus is too thick and remains in the dark. It became clear we needed a method for thinning, a cell. And of course, an obvious attempt was, to use ultramicrotomy, using a diamond knife to cut slices through the ice. But that did not work very well. There were very obvious and very serious artifacts caused by that. One reason for that is that vitreous ice, amorphous ice, is highly compressible. So if you exert some pressure on it by trying to section it physically, you get very massive compression, up to something like 30% or so compression of the sample. So that did not work well, and we had to look for alternatives. Okay, that is just, to make the point again. I think that from then on, I think it got sort of interesting or popular for many people to begin to feel this could go somewhere. Now, we invested then our resources, human and financial, into the development of an alternative thinning method, focused ion beam milling. So again, you see here, a dictyostelium cell sitting on the grid, and we have a dual beam instrument. There is an SEM, so we can monitor what we are doing in the SEM image, and then we have a focused ion beam, in this case, in early times, a gallium beam in order to ablate material, to produce kind of thin compression-free windows into the cell. That is now, again, common practice. So the principle is basically ablation of material by the impact of gallium ions. So we have to reduce the thickness of the sample to make them electron transparent to something, let's say, not more than 300 nanometers. But with that method we avoid, so we can use different geometries. We can ablate material so that we can generate a kind of a wedge-shaped sample with some thin regions, but I think the normal thing is to produce what we call lamella, which are basically slices. We ablate material from above and below the area of interest, and so we generate basically slices or lamella with a thickness typically of, let's say, 200 or so nanometers. So Here come now a few examples. I think that was an experiment done by Julia Mahamid. So we had a HeLa cell sitting on an EM grid on the left-hand top left. You see that is the region in color, that is the nucleus. And here we decided to cut lamella at the periphery of the nucleus at the time. So you see basically here at the bottom, I mean, you see this is a thin lamella, and now we look in the transmission electron microscope onto the lamella. And I think we can now move from inside the nucleus to the outside of the nucleus. So what you see at the top is in the region of the heterochromatin, and you see the nucleosomes fairly easily. You can average them, and I think then you get, I mean, routinely resolutions of something like 4 nanometers, so not yet high resolution, but the problem or the challenge would really be to establish the connectivity between nucleosomes, and that is still, I mean, a fairly challenging image processing problem. As you approach then the inner membrane of the nuclear envelope, you see this meshwork of the nuclear lamina, I mean a fine intermediate filament of something like 3-4 nanometers thickness. Then you occasionally hit a nuclear pore complex, first on the nuclear side and then on the cytoplasmic side. And I think as you get onto the outer membrane of the nuclear envelope, you will see, I mean, many filaments, I mean, actin filaments, etc., etc. Now, then you can do quite a bit of processing. You can do segmentation of all the filaments, of actin, microtubules, of the nuclear lamina, etc., etc. Then, I mean, on the outer membrane of the nuclear envelope, we have many ribosomes, and we can easily average them to a decent resolution. Nuclear pore complex, the structure you see there on the left-hand side is not yet a very high resolution structure, but it's remarkable in that it was obtained by averaging over only a handful of nuclear pore complexes there. And it's important to improve that more by, I mean, boosting contrast, because as you see below, I mean, these are neighboring nuclear pore complexes, and the diameter, I mean, can vary by something like 15% or so, according to different functional states of the nuclear pore complex. Now, what you see now is a synopsis of all the averaging. So again, in the background is the region of the heterochromatin and then nuclear pore complexes. And on the surface, you see ribosomes in the form of polysomes, etc., and actin filaments, etc. So that is an example of analyzing, I mean, a lamellar cut from the nuclear periphery. Now I will show a few data from, I mean, an organism, Chlamydomonas reinhardtii, which has become a kind of a popular model system in our lab. The reason for that is that I think Chlamydomonas cytoplasm and nucleoplasm is a little bit less crowded than, let's say, the cytoplasm of a yeast cell. Moreover, an advantage of Chlamydomonas is it has an almost deterministic topology of its organelles. So that is basically a slice through the entire Chlamydomonas cell, and then you can, okay, so you see, for instance, I mean, here is a region in the box. That is the region around the Golgi complex and the endoplasmic reticulum. So it's, you know where to go if you want to find, I mean, a specific organelle or whatever. Okay, you see the arrow there. Okay, that is, I mean, a gap in the nuclear envelope here. So, and basically that is the region of a nuclear pore complex. We can cut out, I mean, the volume surrounding or including the nuclear pore complex, and then we can do averaging on the right-hand side, and you see in this case we have already a sort of a decent resolution. Still not very high, but something like 3-4 nanometer. Ribosomes are always the easiest to do. So this is now a ribosome sitting on the translocon or the 61 protein conducting channel, and you see resolution is already relatively good. Now we zoom into the translocation of the protein conducting channel, and you see very clearly the helices indicating that now resolution is something like 7 angstroms here. Okay, I should say, and I think here I changed a little bit the topic. We are not really interested in ribosomes. We are interested in the opponent of the ribosome, that is the proteasome. That is the molecular machinery for protein destruction. If a cell wants to get rid of certain proteins in a targeted way, this is something done by the ubiquitin system and the proteasome. The proteasome is by itself largely a non-specific protease. The specificity is conferred by the ubiquitin system, so marking by the attachment of ubiquitin chains, I mean, a given molecule for destruction. Now, the proteasome comes, if you like, in 2 flavors. There's the 20S core, that is where proteolysis takes place. I mean, that is this structure in gold, basically. So, I mean, in the beta subunits, that encloses a central cavity, and that is where the active sites are located. Now, in order to become functional, I think the core complex has to associate with 1 or 2 regulatory particles in light blue. Their role is to prepare substrates for degradation. That includes recognition of ubiquitinated proteins, the removal of ubiquitin chains, because that is a requirement for unfolding. And then, I mean, the substrate has to be unfolded in the regulatory particle and then has to be translocated into the core complex for degradation. Now, I think that is a very low resolution structure. You see, that was 1998, before the revolution, as some people call it. Nevertheless, I think the structure was highlighted in a United States stamp, and one day I got a Christmas postcard from Boston, and I was very surprised to see our proteasome on the stamp. I think even the 20S proteasome, which is an 800 kilodalton complex, was challenging at the time. It was highly controversial. How many subunits, what the stoichiometry of subunits was, etc. So we decided at that point, I mean, in order to be able to move forward with the structure analysis, we looked for proteasomes in bacteria, we looked in archaea, and we were able to find them in archaea, in an organism called thermoplasma. Now, I think as the abstract says there was in thermoplasma a proteolytically active particle with almost identical in size and shape with the so-called multicatalytic proteinase from rat. The term proteasome did not yet exist at that time. It was referred to as the multicatalytic proteinase. I think having found that, I think we were very fortunate, because then we could make proteasomes recombinantly, and we had large quantities of proteasomes, and we could then solve the structure by X-ray crystallography. Now, that is thermoplasma, our organism at the time, and it became, I think, again, a kind of a model in the lab for studying protein quality control and degradation. So we had, in 2000, we had sequenced the genome, and we knew all the components also working downstream of the proteasome. You see on the left-hand side, that was then the structure we got in 95 by X-ray crystallography. It was an effort of something like 5-6 years to obtain crystals diffracting to high resolution. On the right-hand side, if we were doing it today, we would do it by single particle electron microscopy. So that is, on the right-hand side, the EM structure is at a resolution of about 2 angstroms. On the left-hand side, the crystal structure was something like 3.4 angstroms. So whereas the crystallography was an effort of 4 or 5 years, I think doing now the proteasome structure by cryo-EM single particle analysis is a matter of a day or 2. Now, I think the important finding here, but I think there's a little bit getting away from the main topic of my talk, was to find the very unusual active site and a very unusual proteolytic mechanism. Basically, it is a single residue active site. It's the N-terminal threonine that does the job of proteolysis. Now, that immediately offered a way to develop an inhibitor. We did not develop the inhibitor, but we suggested, and I was involved in that, that one could simply extend the N-terminus of the molecule by 1, 2, or 3 residues, and that could be done by using a dipeptide boronate. So the warhead was the boronate, and then I think the N-terminus was extended by 2 residues, and that became a very powerful inhibitor. And as a matter of fact, this inhibitor became a very successful drug. It became a very successful drug for the treatment of multiple myeloma. So unfortunately, I mean, the drug has also unpleasant side effects, but it's still, it became, I think it was a drug development by serendipity. Now, I think we were still, of course, trying to get a high-resolution structure of the 26S proteasome, which is a 2.5 megadalton complex, which has, I mean, the core particle in this region here, and then the regulatory particle. Again, that was before, I mean, the resolution revolution, so the map you see there is an 8 angstrom map, and it could not yet be interpreted in terms of the subunit architecture. That is why you see at the bottom, I mean, we used an orthogonal approach. That is, we use cross-linking and mass spec to have, I mean, distance restraints between the subunits. And in the light of those data, we could interpret the map in terms of the molecular architecture of the complex. Now, the proteasome is challenging and was always challenging. You see 34 different subunits. It's a very labile structure, and it has a high degree of dynamics. Now, I need to introduce the proteasome and the conformational changes it undergoes during its functional cycle to understand the in situ structures I'm going to explain to you. So it was about 10 years ago, we had, I mean, a structure of the human proteasome at 3.9 angstrom, and then we did many studies to understand the conformational landscape of the proteasome. Basically, you see in blue what is docking to the core particle is the AAA module. That, I mean, drives and fuels all the conformational changes. You see the deubiquitination module. You see also the ubiquitin receptors here. It's important to understand basically the conformational changes because, I mean, they are of such a magnitude that even at the lower resolution in tomography, we can infer from the conformation what the activity status of the molecule is. So I think there will be a little movie showing basically giving you an idea of the conformational dynamics. Now that is what we call the ground state, the S1 state, in which the proteasome is not engaged with substrate. It's simply sitting there and maybe waiting for a little bit of substrate to show up. Okay, then there's a so-called commitment state, at which state the process of degradation becomes irreversible, and then there are different substrate processing states. There are rotations of whole modules of subunits in the regulatory particle. Their role is to bring the substrate down from the receptors to the AAA ATPase for unfolding and translocation. There's also a lateral shift that is needed in order to align the unfolding channel in the AAA ATPase with a gate that controls access to the core complex. Okay, that is the dynamics of the isolated molecule. But now, the proteasome has also cellular dynamics. and will be the topic of the next couple of minutes. Response to metabolic shifts is a special topic that is very new work. So, first of all, where are the proteasomes in the cell? And we go back here to in this column here, everything that is in green is GFP-tagged proteasomes. So there are two locations. One is basically they line basically the inner membrane of the nuclear envelope. I think it might surprise you, but a major protein degradation compartment in the cell is the nucleus. Then I think you see one or two very bright spots here. That is, proteasomes sitting basically on the endoplasmic reticulum. And I think there's a well-known and well-studied, these punctae, are highly dynamic. There's de novo assembly, but there's also fusion between them. So basically, that is what is called ER-associated degradation, or ERAD. That is extremely well studied. Basically, what is it about? It is, proteasomes which are synthesized by ribosomes are translocated into the endoplasmic reticulum, where they're expected to fold properly, and then, if they're properly folded and maybe glycosylated, they are exported from the ER in the form of vesicles. But the folding in the ER can go wrong. It should happen with the assistance of molecular chaperones. However, it goes wrong especially under stress conditions. And then there's the so-called unfolded protein response. Since there's no powerful degradation system in the ER, in order to get rid of these misfolded proteins, they have to be retrotranslocated into the cytosol for ubiquitination and degradation by the 26S proteasome. I think there are two players involved that was well known. I think one is, a molecule CDC48, and the other one, is the proteasome. So nothing was known about the organization of ERAD. And so that is something we addressed then by Cryo-ET. So basically, if we zoom into one of those clusters here, what you see then here in blue, these are the ribosomes. I think in different shadings, ribosomes sitting on the membrane and ribosomes that are free, and then in red, that is an ERAD cluster. In red, you see the 26S proteasome plus CDC48. Now, the degradation clusters, the proteasome clusters here, strictly avoid any ribosomes to get there. Basically, this is a condensate, and the condensate excludes the ribosome. So it's all 26S proteasomes. We can zoom into one of those clusters, and then we do what we call a molecule-by-molecule analysis of the tomograms. One can use template matching. So, for a structure, if we have a molecule whose structure is well known, we can use that as a template and look for corresponding structures then in the tomogram. That is not what we have done here. What we have done here is basically we have picked all the densities in the cluster, did a sort of a classification according to similarity, and ended up with two subpopulations here of particles in the cluster, and we can do subtomogram averaging of the two clusters here. But we keep the information where all the molecules going to this cluster or this cluster came from in the original tomograms. Now, that is the result of this kind of analysis. So the cluster is roughly here. You see, in red, again, the 26S proteasome, in yellow color, CDC48. So then I think you can do a deeper analysis. You can do a classification of all the 26S proteasomes. You can say which molecule is in what assembly state, 1 or 2 regulatory particles. You can also, determine whether it's in the ground state or whether it's in the substrate processing state. Now you can look at those in the substrate processing state depending on the distance to the endoplasmic reticulum membrane. If you do that here and look at those that are very close to the the ER membrane, within 20 nanometers or so, then you see that their very large fraction is actively degrading, in a substrate processing state. When you take those now and do subtomerium averaging, then you see attached to the regulatory particle, some density which is not accounted for by the proteasome itself. So tentatively, this was interpreted by us as being substrate without having real proof. Now that was a surprise because what people expected was to find CDC48 to be, next to the membrane, to do the retrotranslocation of the substrate, and then to hand over the substrate to the proteasome for degradation. But we see CDC48 at the periphery of the cluster and not, directly attached or in the vicinity of the membrane. So we decided there must be something or two different mechanisms. One we call indirect ERAT, that is, translocation by CDC48, and then degradation, handing it over to the proteasome. Versus, what we call direct ERAT, that is basically, that the proteasome is involved or does the retrotranslocation, pulling it out of the endoplasmic reticulum and then degrading it. Now, that was a little bit against the dogma, but there was other evidence that had been ignored by the community. There was, for instance, a paper by my late colleague Stefan Jentsch saying the proteasome in membrane extraction of short-lived ER proteins, membrane proteins is involved. Another paper by Michael Glickman at the Technion in Haifa, also the role of proteasome in membrane extraction. So there had been biochemical evidence for that, but it was not the prevailing view. The second group of proteasomes is, as I said before, basically in the nucleus. So this is the nuclear envelope here. Here's the cytosol, a mitochondrion. And then you see, nuclear pore complexes. If you map back all the proteasomes, that is those in light blue, they're everywhere, but we can distinguish those in the ground state, and we can also classify them according to their interaction partners. This is a nuclear pore complex, and we have quite a high density of proteasomes near the nuclear pore complex. But if you do a deeper classification, there are two groups of attachment sites. There are those, basically very close to the membrane level, located at the periphery of the nuclear pore complex. And there's a second population in yellow that is here. These are at the site where the nuclear basket is located. The nuclear basket is a fairly flexible structure. It does not show up very well in the subtomogram averages. Now, okay, I don't want to go into quantitative detail here of the classification. So we have two types, probably interactions with the nuclear pore complex. Some here at the membrane, probably their role is to maintain the difference in protein composition between the outer and inner membrane of the nuclear membrane. And then we have another population that deals, with stuff that is coming into the nucleus and removes misfolded ones. I come to a different topic. We had, back almost 10 years ago, a major project called TOPAC, Toxic Protein Aggregation in Neurodegeneration. It was a collaboration between the labs of Ulrich Hartl, who is interested in proteostasis networks, Matthias Mann, an expert in proteomics, so he wanted to find out what is the cellular proteome change in response to toxic aggregation. And our role was, what is the in situ structure of aggregates? How do they interact with the cellular environment? Now I'm talking a bit about the work of Shang-Wu, who is in the audience today. So I think one was, basically a neurotoxic aggregate that is responsible for ALS or FTLD. So terrible diseases. So basically the hallmark is a hexanucleotide expansion in this gene here. And that results in the synthesis of toxic D-peptide repeats. And what is found in the brains of patients suffering from those diseases is basically glycine-alanine repeats. Now, glycine-alanine repeats, I mean, have probably a silk-like architecture. Some viruses use, I mean, this kind of repeat in order to escape degradation. Doing that, this kind of project, I mean, requires a relatively complex workflow. So the first step is always, of course, vitrification. I mean, getting to freeze the cells rapidly. Then I think the cell is a large field of view compared with the field of a large field compared with the field of view of a tomogram. So we must make sure where are the aggregates located. That is done by fluorescent light microscopy. We transfer the coordinates then to a FIB machine and can cut a lamella that includes then the aggregate material. So that is the grid here. Here you see in this, here is a cell, and it's highlighted in green that is a cell that carries, I mean, an aggregate. So we cut, I mean, lamella, including the aggregate material, and then we can take a tomogram. That is one of the tomograms Zhang has taken at the time. It looks at a glance a little bit like these are fibrils, but these are not really fibrils. I mean, as you do now a segmentation of the aggregate material in red, you see they are more like polymorphic ribbons. But the interesting observation here was that basically there's only a single molecular species inside the aggregate material, and that is in light green, I mean 26S ribosomes. All other macromolecules, including ribosomes or chaperones, I mean acting downstream of the ribosomes, I mean are excluded from the aggregate material. But we have a fairly massive increase in the concentration of proteasomes inside the aggregate material. So because of the sheer number and the abundance of proteasomes there, we could get a fairly decent resolution structure of the proteasomes there. In green, you see proteasomes that were in the ground state, and in blue, you see those in the substrate processing state. Those in the substrate processing state, we also see again, I mean, here in pink, an additional density not accounted for by the proteasome itself, and again, tentatively interpreted as substrate. But to show that more clearly, if you map back now, the blue guys, the substrate processing ones, they almost invariably are in direct contact with the aggregate material. The interpretation is the proteasome is making an attempt to degrade the neurotoxic aggregate, but the characteristics of the glycine-alanine repeat does not allow the proteasome to unfold. I mean, it's too expensive to unfold them, so the proteasomes become stalled. And that compromises, of course, the protein quality control system of the cell. The second system was, I mean, Huntington's disease, and that is a terrible disease affecting coordination, cognitive decline, etc., etc. Now, again, the same workflow. Okay, I think what you probably know is, I mean, the characteristic of Huntington is a polyglutamine extension here in the structure of Huntington. That is also a structure done by Zhang at the time. And that results in an unstable protein, which, I mean, tends to form aggregates. Here we zoom now into a Huntington aggregate. So you will see basically in blue, these are the Huntington fibrils. And then you see in red, these are remnants of the endoplasmic reticulum. Basically, whenever the filaments touch the membrane changes its curvature, and it becomes completely destabilized, and so as a consequence, the endoplasmic reticulum here becomes highly fragmented, and the ribosomes sitting on the ER are liberated and sitting here in the remnants of the endoplasmic reticulum. I think these two studies from from our lab were sort of commented in a News and Views by Wade Harper's lab at Harvard Medical School, so saying, well, very different mechanisms here. I mean, destruction of the membrane system here, compromising the protein quality control system. Now, I think that will be the large proteasome story here. That is some very recent work. So I think it's basically based on the following observation. Proteasomes are great consumers of ATP. Just to give you an idea, to degrade a medium-sized protein, I mean, it requires something like 80 ATP molecules. That has to do with all the conformational changes. I mean, the opening of the gates of the catalytic core, the unfolding channel of the AAA ATPase, so everything, I mean, is consumed here. Very costly for the cell. So the question was, what does the cell, under conditions of ATP shortage, in order to shut down the consumption of ATP by the proteasome? Now, there was an observation. Basically, under conditions of starvation, here in budding yeast, the nuclear proteasomes are transported into the cytoplasm to form puncta, which are referred to as proteasome storage granules. So that is what you see here. Basically, you see in green these puncta here. These are now the proteasomes suddenly in the cytoplasm in the form of these PSG puncta. So the question, at the beginning of the experiment was simply, what is the molecular organization of these puncta? I think it was known that, in these proteasome storage granules, the proteasome is not active. But the idea in the field was it's not active because basically the core complex and the regulatory particle had fallen apart, had dissociated, and so then the machinery becomes dysfunctional. But the question is, was that true? So we began, to work from, the beginning of, that is still, at a stage of proliferation. The cells is growing. And then I think as we approach, if we go into glucose starvation, the onset of starvation, I think things begin to change. What happens is, and it's very surprising, the observation, suddenly the 26S complexes form a supramolecular assembly, a trimer of 26S proteasomes here, which is originally, or in the beginning, still dispersed in the nucleus. But then, upon longer starvation, at a couple of hours, the trimers of the proteasome, they congregate here at the nuclear envelope. What you see here on the right-hand side is now, this is now the structure at about 8 angstrom of the proteasome trimer here. And we can very clearly see that each 26S complex is in the S1 ground state. And by forming the trimer, basically the movements in the regulatory particle cannot continue. It becomes completely stalled or arrested in the ground state. It's an interesting way of inactivating this ATP consumer. Now the next step is then they move to the vicinity of the nuclear pore complex for nuclear export. And the trimers then leave the nucleus and they accumulate then, some of them are also, tethered to the nuclear envelope by a very filiform and flexible tether, which we have not yet identified. The molecular identity is not yet clear. It's too flexible to show clear density. We see only a density, the site of attachment to the regulatory particle. Now, how are the PSGs formed? Basically, the trimers, generate long fibers by assembly in a linear fashion. And then the building block is very clearly still, the proteasome arrested in the inactive ground state. So the same conformation as early in starvation in the nucleus. Then I think they assemble further. I think they begin to to align with each other the fibers of trimers, and that is again a subtomogram averaging now, average of the trimer in the fiber. The structure is again unchanged, but then they align and form bundles. Now, it's a typical feature of paracrystals that, it's not a perfect crystal, so we have two types of bundles. We have 5-membered bundles and we have 6-membered bundles. So it's not a perfect crystal at all. I think in the beginning, when forming these proteasome storage granules, if you do experiments by photobleaching, FREP or FLIP, whatever, this is, a kind of a liquid or fluid kind of condensate. Now, over time, the storage granules solidify, and the solidification, is the result, of course, of paracrystal formation. What we can do is the following. We can look more closely for the interaction within the trimer. And of course, all the interactions are in the regulatory particle. That is why everything becomes kind of stalled or arrested. It's very clear that the main subunit making the contact is a subunit called RPN9. You see that more clearly here in this ribbon diagram. So RPN9 is this subunit here, and that is sitting here at the interface. There's also a kind of a peptide, SEM1, that also contributes to that. So we asked the question, what happens if we inhibit trimer formation? And we did that first by a fairly brutal mutagenesis. So we deleted the N-terminus of RPN9, and then we did many point mutations, in the interface region. What happens is, they no longer formed trimers in the nucleus, and they did not exit the nucleus anymore. They were retained in the nucleus. There was no formation then of PSGs. Now, what happens if we restore, glucose, and so the cell can produce ATP again? Then what happens is basically that one after the other of the 26S proteasomes adopts a conformation different from S1. So, and then, if one or a few of them adopt different conformations, the paracrystals become unstable. This is, illustrated here. Basically, like, each card is one 26S complex, so if one card, changes its conformation, the whole thing becomes unstable, and the proteasome storage granules all fall apart. Basically, that is the summary of this story here. So we have at the beginning of starvation, we have the trimer formation. The trimers then leave and they go into a liquid-like granule here, which becomes solid and paracrystalline, and until it falls apart when the cell resumes growth and it goes back to the nucleus. It's a fairly elegant mechanism. I think in the last few minutes, a little bit about maybe the future. I think that is from a review which I wrote with my former graduate student, Martin Beck, now director at the Max Planck Institute in Frankfurt. The question was, can cryo-electron tomography reveal the molecular sociology of cells in atomic detail? Not yet really, but I think with the exception maybe of ribosomes. But I think there are a couple of things we can do to improve cryo-ET to realize its full potential. Throughput is a limitation. I think data acquisition is no longer a limitation. In the past, I think we were happy when we got 3-4 tomograms per day. Now I think it's maybe 300 per day. Sample preparation, especially FIP milling, I think is still slowing down things a little bit, or still slowing down, and I think the workflow is not as well integrated. It requires still transfer between instruments, so that is something that should be improved. I think the most important thing is still to improve primary data quality. I think the detectors, the direct detectors we are using these days, they are not yet, still don't have a perfect detective quantum efficiency. I think the biggest hope in the field is that an improved faceplate, the laser faceplate developed again at Lawrence Berkeley Laboratory, and I think installed also at the Zuckerberg Imaging Institute in Redwood City. I mean, now I think there are several developments. There will be new generations of cameras from different manufacturers, I mean, going now close to the physical limit. But the laser faceplate shows huge prospects to boost contrast massively. Unfortunately, it comes with an enormous price tag. An instrument with a laser faceplate will be $15 million. I think another shortcoming is the coverage of cellular space. I think, as you can imagine, I mean, these thin lamella accommodate only a small volume, small compared with the volume of a cell, maybe a few percent or even less. How can we improve on that? I think one is, of course, I mean, serial lift out, there's no time to go into that, but I think that can improve the coverage of cellular space quite a bit. But I think it will also be important to develop an alternative technique, and that is cryoplasma FIP volume imaging. There are only a few preliminary experiments, but again, there are serious efforts at several places right now. Yeah, I think the biggest challenge is improving data interpretation, because in tomography, we see not only what we are looking for, we get the whole picture, but that comes with opportunities, but also challenges. So we have, in the end, millions of densities or billions of densities in a cell, and we need to identify to which molecule, what is the identity of the molecule in these density maps, and we have to annotate that. So there are a couple of possibilities to do that. Again, I don't want to go into details. Maybe one, okay, that is just, I mean, an idea. FIP milling has been improved, speeded up by plasma FIP milling, and also there's a lot of automation on the hardware side, but also a lot on the software side. So this has become pretty fast. That is a first example of volume imaging, in this case of a, I mean, I don't know whether you know what volume imaging means. You remove, let's say, a layer after a layer, and then you take an image of the face from which, I mean, that was removed. So basically, the material that resides there. So that is, I mean, something I think that should be developed. I mean, volume imaging exists, I mean, in connectomics, but I think that is not in cryo. And if we want to make the two methods and want to integrate them, we have to go into cryo. So the next frontiers, that is the last slide. So regarding outlook, I think we have to move from currently looking, let's say, at one molecule like a proteasome in the cell to, I mean, a comprehensive visualization, identification, and annotation of all densities, basically of the entire proteome of a cell. That is, of course, a huge challenge. So getting, I mean, holistic images of the entire cell, that is not the challenge. The challenge is then the interpretation here. So the identification and annotation. So I think this is, again, something from Jean-Guo's work and from a collaboration with Axel Brünger's lab. I just want to make the point, I think one strategy is, which is here done only for one molecule or for one density, that is from a synaptic vesicle. The big molecule here is easily recognized by its shape, that is a V-ATPase. But then there was an observation here, there's an additional density here in the vicinity. So what is it? How can it be identified? And I think that could be a kind of a paradigm of how it can be done. So basically, one has to have a high-resolution structure that can be compared with the density here in the sample. So in this case, it does not need to be an experimental structure. It can also be an AlphaFold prediction. And so we can then, I mean, use the AlphaFold model and I think score the similarity to this density. In this case, it was also relatively simple in the sense that there were very few candidates, but these candidates were very similar. So that was a challenge. So basically, there are paralogs. So, and it's clear that I think if you score the similarity, you minimize the outliers where, I mean, the predicted high-resolution structure does not fit the density, you want to minimize that. Now, that has to be scaled, and I think that is where machine learning can probably help a lot. Now, finally here, that is my final comment here, is a citation from the work of a famous mathematician, Freeman Dyson. New directions in science are launched by new tools much more often than by new concepts. The effect of a concept-driven revolution is to explain all things in new ways. The effect of a tool-driven revolution is to discover new things that have to be explained. Thank you very much for your attention. I think I should mention I mean the collaborators of the projects that were carried out in the past 5 to 10 years or so. I think if I would, the complete list of people who contributed to the development from early on, I think that would fill the screen. Okay, thank you for your attention.
Thank you, Professor Baumeister. Please take a seat at the stage center for the question and answer session. May I also invite Professor Kang on stage to moderate for the Q&A session, please?
So I should moderate Q&A session. Thank you again for your excellent talk and the early history as well as insight for the future. So yeah, I really enjoyed the talk. Yeah. Okay. Questions from audience are welcome. Please raise your hand for question, and our staff member will hand over the microphone for you.
Hi. It's really an honor to listening to your talk in Shaw College here. And I'm a student from Shaw College, majoring in religious studies, and I'm also really interested in life science and medicine. And I have a little question here, is that given that the advances in cryo-FIB milling and the AI reconstruction technology, how close do you think we are to having the fully comprehensive visual protein of a eukaryotic cell? Thank you.
I'm not sure I've completely got the question. I think it was how AI would influence things. I think it will come primarily on the image analysis side. I think the last example I showed, I mean, basically, I think in a way, AlphaFold prediction is already, I mean, of course, a product from AI to some extent. I think there are also, at the other end, real efforts amongst the manufacturers to use AI to optimize the data acquisition. I mean, to have, setting the imaging parameters of the microscope to an optimum. So far, the results there are not yet very impressive, I have to say. Basically, I think an experienced microscope operator is still a bit better in setting, the imaging parameters to extract the maximum of information. But I think that might change. I think last time I have seen the efforts is a bit more than a year. At that time, it was not advanced at all.
Thank you. Next question. Somebody.
I have two questions. So, first is a failure always happens in scientific research. Just like yesterday, I ran a PCR gel and there's no result. So I was asked myself, am I really suitable in scientific research? I know I shouldn't have these feelings, this thinking, but sometimes those emotions may happen when you're facing failure. So do you have any suggestions to deal with those bad emotions or bad attitude towards failure?
Can you repeat the question?
So when we do experiment, there are a lot of failure, especially for cryo. There are a lot of bad samples that may not be imaged by tomography and high quality data.
Well, yes, there are always, of course, problems, and I think disappointments. I think still a major problem is where we have to invest. I think vitrification is usually not perfect. I think as we go, and we have to go to larger samples. I mean, to get away from single cells, we have to go to organoids or something like that. And I think then we still have quite a bit of ice crystal formation. And yeah, we are trying to launch programs to come up with, I mean, even if you use high pressure freezing, which makes, of course, the workflow again a bit more complicated, because not the high pressure freezing itself, but I mean to make it easily compatible then with targeted VIP milling. Plant cells are notoriously difficult to vitrify.
Yeah.
I mean, because of vacuoles and other stuff. So I think if you are working on plant cells, you will have many frustrations because of incomplete vitrification. I think we hope that there will be, I'm encouraging the Chen Zuckerberg Foundation to launch a kind of a bottom-up program to come up with better ideas. The problem is that even high-pressure freezing was developed something like 50 years in the context of freeze etching. So we are using here technology that is completely outdated, basically. So, but there are many other, I mean, I think you have to accept that when you do experiments, they can go wrong and there will be failures, okay? I think that is where the quality has to be persistent.
Thank you. And my second question is, I'm also studying cell and molecular biology, and I'm very interested in stem cell neuroscience and cancer research. But do you have any advice or suggestions to the scientists of our generations? So, just for example, like what we should focus on and what we should be careful.
Yeah, you should focus on where your curiosity drives you. Okay, I think the starting point has to be curious about something. And I think if you are truly curious about something, then you will also become passionate to solve the problem. And I think then I think also maybe outside the box thinking is an important thing. So you should not be limited or confined by what is out there, what is going on, what is the trend. I think you should try to come up with completely fresh ideas. Okay, so it's easy to say, but I mean, I think in a way, maybe I should not say it in that way, it's an advantage to be ignorant to some extent. If you're ignorant to some extent, I mean, then you will automatically do outside the box thinking, okay? You will not be limited by, I mean, what other people have done or suggested or proposed or, yeah. I mean, there's no simple recipe to success.
Yeah, I think your pursuit of cryo-ET is an example of thinking outside of the box.
Yeah, okay, I think, yeah. So you should not always listen to advice. Yeah. I mean, I think the advice would have been, don't do cryo-ET.
Yeah. Thank you so much.
Hello, Professor. Is this tomography is replacing MRI or they work together?
So it's cryo-ET can work together with other like solution-based technique like MRI.
Yeah,
And MRI.
I think, well, you want to do imaging on different scales. So I think I see a great future for volume imaging in order to get an overview of large samples, large cells or tissues, etc. And then I think there are ways of integrating the high-resolution information coming from tomograms. And I think I'm a strong advocate also of integrative approaches. I mean, taking advantage of orthogonal methods. I mean, for instance, I mean, when you do what I described at the end, something like template matching, the outcome of that will never be error-free. So you have to validate And I think for that you need orthogonal methods, and I think one of the most powerful ones is cross-linking and mass spec, so that gives you hard numbers about the distance between two molecules, and that can either confirm or reject basically the result then of a method such as template matching.
Okay, hello, Professor. So I'm a student come from Professor Kang's lab. So I have a question which I'm very curious about. So if we can use this kind of the structural analysis to solve a very large scale structure, for example, so we all know that in our body, so for some neural cells, so it maybe can go over for 1 meter for 1 axon in the single cell. So if we want to just to localize this very, very long cells, so can we use this such as some volume EM or some Fib analysis, or this Cryo-ET nowadays to solve a very big, very large
Well,
structure?
a meter is very large for tomography. I mean, that is,
yeah, actually, yeah.
I think that is not easily possible. I think the only way is then, to take a series of tomograms and to kind of tessellate them together. To make a montage of tomograms. These methods are relatively well established, not yet in cryo-ET, but in conventional electron tomography. I think there you can tessellate, tomograms, I think they are pretty good methods. Piecing it together from many separately taken tomograms. I think that is the only possibility I see there. But a meter is maybe, you should be more modest. And
Yeah, I think 1 meter too ambitious.
Thank you very much.
Yeah, thanks for your talk. And I have one question about the quality of the cryofixation. I was wondering if there is any standard method to define or to understand the quality of fixation, besides of the visual check of the lamella, the how to say, the look of the lamella generally under TEM. Is there any quantified method to know that the quality?
Problem with the acoustics, so I need an interpreter.
I think what she's asking is she does like plant cell high pressure freezing and cryo-fit. And is there any like a quantifiable measure that whether you want to do proceed with the cryo-ET or like give up?
No, you see relatively easily whether you have ice crystal formation, okay?
Right.
So you can, of course, exclude that, but better, you don't have ice crystal formation in the first instance, okay? But I think we need desperately, better technologies for vitrification.
Okay.
I think there are ways, of course, to try to do that, and I think people in the lab have tried to use cryoprotectants, to some extent. That works, reasonably well with some animal cells. Let's say 5% of glycerol, I think you can do, but these are all measures that were also developed, in the 50s, 60s for freeze etching, but it's a bit dangerous. In plant cells, I can say, I think we have never been very lucky with glycerol or something like that. There are also other compounds, but I think we need better physical techniques for vitrification.
So yeah, you do better work on some other like non-plant cell maybe.
Hello, Professor. Thanks for your talk. I have a small question about your technology. If we give an electron to a molecule, this molecule will be charged and related to other geometry. If we use electron to do this imaging task, where the protein or other objects in cell will change, and let me organize my language. Okay, if we use electron to do the imaging task, where electron influence the structure of these molecules.
So the electron imaging, whether they affect the structure in the cell.
Pardon?
Electron imaging, imaging the structure.
Yeah, of course. I mean, exposure to electrons, of course, causes radiation damage. I mean, so you have to keep the dose as low as possible. I mean, that is, I think, basically in the 60s, 70s, I mean, low-dose procedures were developed. I mean, the only real solution there is, I mean, to go to very low doses and to restore signal to noise, I mean, afterwards by post-acquisition image processing in the form of denoising, subtomogram averaging. Subtomogram averaging can be very powerful, but I mean, it has limited, I think, to larger molecules, and it's limited by abundance. I mean, the difference in single particle analysis is you have no limitation with the number of particles because you concentrate them. In tomography, I mean, the abundance of particles is determined by nature. Okay, so if you have, I mean, we had molecules we were very interested in, but we had maybe something 1.5 copies of the molecule per tomogram, okay? Then it becomes very hard, I mean, to do subtomogram averaging. Then you have to take hundreds of tomograms in order to get anywhere. So, no, I think there are, of course, other damaging mechanisms. Also, FIB milling is not completely artifact-free. I mean, in the beginning, we thought it was just the implantation of gallium ions at the surface, so, but I think that did not matter because, I mean, if you have a lamella that is 200 nanometers and you have, let's say, gallium in the first 5 nanometers, okay, so you simply cut away that, okay, that is not an issue. Now it is becoming clear, again, from kind of template matching that the damage is a little bit deeper beyond, I mean, implantation of the gallium ions. But I think it's still, the damage by FIB milling is relatively modest. So if you take, I mean, a specific molecule, let's say a ribosome, let's say 10 nanometers from the surface, and then I think a bit deeper, I think there is damage. I mean, the quality goes down, but it's fairly modest, okay. So it's radiation damage is, of course, was the biggest problem always. I mean, because of the need of multiple exposures.
So, any other?
When freezing the cell, will the ice affect the result? Like the ice inside the cells?
No, if it is truly vitrified, it will not affect the result. But I mean, if it's crystalline, of course, then I mean, it causes damage. I mean, that is, I mean, something one has to avoid. It's easier to say than to do.
Maybe, can I ask a question?
Sure.
So, your template matching, so needs some special, like easily identifiable structural feature. So in your case, you did proteasome particles, and Martin bag is a component of the nuclear pore complexes, and like a rasparosome by Ben Engel recently. But is there any other low-hanging fruit that you can do template matching to identify and then sort and do particle averaging?
We introduced template matching something like 20 years ago, and I think we tried it with what we call phantom cells. So basically, we created lipid vesicles enclosing a well-defined set of macromolecules, and we knew the molecular ratios of the different molecules in, and it worked quite well. But as I said, it's not error-free, so I think it's okay because you might not have, first of all, a perfect template, okay? So I think you might have a high-resolution structure of the molecule you're interested in, but it might be in a somewhat different conformation in isolation, or in the way it was studied, let's say by crystallography from, the in situ conformation. So I think it will never be an error-free method, so it will always require additional validation. Okay. But I think it's the only method that can be scaled to, identify a very large number of densities.
Okay, I see.
And as a matter of fact, labeling, I think, is also very tricky. Even many genetically encoded labels lead to perturbations. I think if you want to develop them, in order to have higher contrast, so you have some metal, that is very often poisonous. Or, you have, a nice label, but it might go into aggregates. So, then it is no longer in the position where you hope it will be. So, I think the fact that there are so many papers, describing always improved labels tells you already that the existing methods are unsatisfactory. So I strongly believe the future of identification is template matching, in spite of the risks.
Thank you, Professor. I'm a science student, and I'm a little bit curious about, you mentioned that kind of protein, they are used to disorder the protein that's formed, right? But why they stay in the nucleus? Because nucleus is not the place protein form, right?
You are asking why proteosomes are so abundant in nucleus? Oh, yeah, he's asking, yeah.
Well, the why questions are always difficult to answer in biology. I think it is a compartment, I think, but that is well known from other studies I know. People like Susan Gasser and etc., they worked on it quite a while. It seems a bit strange that the nucleus is a degradation compartment, but I think the evidence for that is very, very strong, not just from tomography. So, well, it's after all a compartment, I think, with a well-defined mechanism to get into and out of. So, I think many other organelles or so, it would be very challenging to get the substrate into that location. So in the nucleus, there is a nuclear pore, and I think you can be tricky and to use that to get things into and out. So, there's a pretty clear transport mechanism.
Thank you so much, professors. And now may I invite Professor Freedom Leung, head of Shaw College, to present our token of appreciation to Professor Baumeister and Professor Kang. So first, we'll have Professor Baumeister, please. Thank you so much, professors, and may our honorable guests leave the auditorium now. Thank you all for joining us, and we hope you enjoyed the lecture today. Have a nice evening, and goodbye.